very good morning to everyone. Myself, Dr. Prachi Vyas from Faculty of Pharmacy, take this privilege to warmly welcome you all. Your presence truly enriches our gathering. This is the session first of third international conference of integrative chemistry, biology and translational medicine, ICBTM 3.0. The core of invited lecture is biology of infectious disease and drug discovery second. Now, I would like to invite the chairperson, Professor Aniko Borbas, Ma'am, University of Debrecen, Hungary. Professor Aniko Borbas is highly respected professor at the University of Debrecen in Hungary. Her prolific career spans over numerous contributions to academia, with more than 200 articles published in esteemed journals. Her work has gathered over 2,000 citations, showcasing the significant research within her field. Now, now I would like to request Dr. Maya Sharma to felicitate Professor Aniko Borbas. Now I would like to request Dr. Preeti Kumar to felicitate Professor Peter Prizer, MIT, SMART, NTU, Singapore. Now I would like to request our chairperson to lead the plenary lecture.
Good morning to everyone. Good morning to everyone. It is my great pleasure to introduce Professor Peter Preiser. He's chair in biological sciences at Nanyang Technological University. And he has held several key roles at NTU, including Associate Vice President from 2019, Chair of School of Biological Sciences, and Director of NTU Integrated Medical, Biological and Environmental Life Sciences. And his research focuses on understanding malaria parasitic evasion of host immunity and drug resistance mechanism, as well as identifying new drugs and new targets. He serves as co-lead investigator for the Antimicrobial Resistance Interdisciplinary Research Group, a program funded by NRF, and his goal to address antimicrobial drug resistance. The stage is yours, Peter, please.
While they load the slides, I'd like to thank the organizers very much for the invitation to come here. It's been a fantastic couple of days. I think the hospitality is amazing, the science is very inspiring, and it gave me the opportunity for the first time to come to the northern part of India and, just learn a little bit about the country. Okay. What I'll be talking about today is about really an aspect of the malaria parasite that's only recently has gathered or gained some attention, mainly about the epitranscriptome. And Professor Leska yesterday gave a nice introduction about why malaria is still an important problem, so I will skip this. So the main thing I will just highlight is that the malaria parasite has a really complicated life cycle involving two hosts, involving both sexual and asexual replication, and also involving a developmental cycle inside the red blood cell, which is similar to a developmental cycle in more complex organisms. I'll be mainly focusing on the erythrocytic stage, the blood stage cycle, but I will also allude to the sexual stage of the parasite, mainly the gametocyte development. Oh, it needs to be connected to the computer to work. Okay. All right. So now it works. So just because I think not everyone is familiar with epitranscriptomic, my talk will focus a little bit on an introduction. What is epitranscriptomic? Why is it important in the context of Plasmodium? And then gives three specific examples of how epitranscriptomic plays a role, and maybe if there's time we can think a little bit about how these mechanisms can actually be an opportunity for new interventions against the parasite. So I think you're all familiar with the central dogma: DNA transcription leads to RNA, RNA leads to translation and leads to the protein. You may also be aware that there is an epigenetic mechanisms where modifications of the DNA lead to changes of the expression, changes in the RNA modifications lead to some changes, And then obviously modifications of the protein also have functional implications. But in the context of RNA, you would think about, well, there is AUGC, but when you really look carefully, there is a lot more, because there are over 150 different modifications of RNA that have been recorded so far. These modifications happen post-transcriptionally. They are quite complex in some cases, and they involve a significant investment by an organism to actually put these modifications in place. Just to give you an example, these modifications can both happen at the sugar, I don't think the pointer works, but in the red sort of as well as the nucleoside. They can be very simple from the perspective of just a single methylation, to more complex modifications, which often involve multiple enzymatic steps to get there at the end. Okay. Modifications happen in all RNA molecules, ribosomal RNA, non-coding RNA, but the RNA which is most modified is actually the tRNA, where more than 100 different modifications have been recorded in different organisms. So how does this actually work? You have to think about it. To put a modification on, you need a writer, an enzyme that puts these modifications on. You also, in some cases, have something which takes them off, an eraser. And then you have molecules or RNA binding proteins which actually recognize these modifications, and through that process have certain biological impact. The impact on RNA in relation to these modifications are wide-ranging. They have an impact on stability, processing, they have an impact on translation, the localization, and even further subsequent modifications. In addition, they also have an impact on the proteins that might recognize these modifications. They impact their function, the interactions of this protein with other proteins, stability of the protein, as well as the localization. So there's really a wide-ranging impact these RNA modifications have. In terms of the biology, what we know about it, for example, modifications in tRNA have an impact on how tRNAs are able to translate different codons, so codon usage, but they also have an impact on the tRNA biogenesis, with the lack or the presence of certain modifications are associated with the cleavage of tRNA, leading to these tRNA fragments, which themselves become regulatory in terms of regulating the translation of certain transcripts. Modifications on the mRNA have been shown to be associated with either stabilizing or leading to accelerated degradation of messages, and then therefore again leading to a transition from one cell stage to the next. So what about epitranscriptomics in the malaria parasite, and why is this particularly important? And I will focus here today in the talk mainly on tRNA modifications, not on some of the other ones. So, as I said, parasite has a complex life cycle, and when you look at the blood stage life cycle, what you see that from a transcriptional perspective, there's a very tight association between every gene and when it is transcribed. So every gene gets transcribed once during this developmental cycle, and what's known is that in the malaria parasite, transcription regulation is relatively unflexible. There are very few transcription factors. What's also known is that a lot of times, for a lot of transcripts, you have a peak of transcription and the protein actually is delayed significantly in terms of when it actually is expressed. So there is some kind of translational repression in the between transcript and translation. Some years ago, we investigated this more closely, and what you could see is that there is very clearly a very low correlation between the transcript level and the protein level, and there's a huge variation of how that correlation exists, with some being significantly delayed. In some cases, there's a lot of RNA and very little protein. In some cases, there's little RNA and lots of protein. And so understanding how this translation is controlled was really the focus of, or it would suggest that there's a significant role in translational control after transcription. So what do we know about translation control in Plasmodium? Well, there's some idea that mRNA turnover, antisense transcripts play a role, that translation initiation also is a key factor, but that there's also a translation repression. So we focused really on the role of the tRNA and translation. And just to give you an idea why this is important, tRNA, I said, is highly modified, and the modifications which are in position 37 and 34 of the anticodon loops have been implicated in many other organisms as being key factors that contribute to what codons tRNAs can actually effectively decode. And so we are therefore also, well, maybe what is also known that in yeast, for example, a specific modification at a specific tRNA leads to the translation of a particular codon-enriched mRNA, while the ones that have the synonymous codons are repressed. So you can, by changing one single tRNA modification, you can actually impact the translation of different transcripts that have different codons. And that's really the question that we were asking, is that something that a malaria parasite also uses? And now, just to give you an idea to study this, it's a pretty complicated platform. You need to be able to identify and quantify modifications, and that's a mass spec-based approach. You need to correlate this or relate that to translation as well as to transcription, to then integrate the relationship between the modifications, transcript level, and protein levels. So the first thing we did was actually to set out to just identify what modifications can we identify in the tRNA. And at this point, we have clear evidence for 25 different tRNA modifications in the parasite's tRNA. And when you look how these tRNA modifications change over time in the developmental cycle, what you will see is here, this is early, this is the late stage of this blood stage cycle, that these tRNA modifications go from low to high in some cases, and in some cases from high to low. So there is a dynamic change of these modifications over the life cycle. So what is the relevance of this? So what happens as a parasite matures in a red cell, it changes, it upregulates its metabolism. There are certain particular genes that are being needed for the final development, segmentation of the chysons, and so on. So there is a need for a stage-dependent translation of certain transcripts. So how do you then actually address this question and how is that related to the modifications? And we focused in is particularly 3 modifications which we identified in the parasite tRNA, which are known to be only associated with the anticodon loop and are specific for the decoding capacity of a particular tRNA. Particularly, these modifications are, if you have these modifications, you preferentially decode these 4 different codons for glycine, arginine, glutamine, and so on. So, knowing this, just to revise, with the presence of the modifications, what you would, in the absence of the modification, this tRNA would be able to, the glycine, decode all these different 4 codons equally, while in the presence of the MCM5U modification, it will only or preferentially decode only this particular anticodon. What that means is that messages which are enriched for this codon would be made more effectively in the presence of the modification, while those would be repressed. So, as I said, these modifications are upregulated, and so the question is, are proteins that are highly translated in the late stages, when these modifications are present, enriched for particular codons? And so the way we did this, we did a quantitative proteomics, so we looked at the most highly expressed proteins at a particular stage, and then we looked at how their codon usage pattern. And what you will see is that proteins which are translated early, or highly expressed early, are enriched for this codon, while protein which would be decoded by non-modified tRNA, and proteins which are translated late or highly expressed late are enriched in the concordant codon GGA, which is specifically or preferentially translated by the modified tRNA. So that's consistent with the idea that these modifications play a role. You can now extend this study by doing sort of more statistical analysis to see what happens when you look at codon usage in the late stage versus early stage proteins, and you see that the late stage proteins have a different codon usage than the early stage proteins. So it's not just the ones that we looked at, but overall there's a clear statistical significant difference in the codon usage of early and late stage proteins. You can then ask also, well, okay, that works for early and late. What happens in the late stage where you have this modified tRNA? Are proteins that have the other codons repressed or poorly translated? So we looked at the highly and lowly expressed proteins. Sorry, no, I went the wrong way. In the late stage, and what you see there is indeed proteins which are highly translated are again having enriched for this GGA. Proteins that are poorly translated have this anomalous codon. So that is really consistent that the tRNA modifications do indeed regulate the translation of codon bias proteins in a stage-specific manner. And you can look at this more extensively, again, by looking at the codons that are used in the upregulated versus the downregulated, and they tend to be synonymous codon pairs. So that for the same amino acids, codons that are enriched, there is a specific codon that is used in the upregulated versus downregulated protein. So what that suggests is indeed that the reprogramming of tRNA modifications does have an impact on the translation of stage-specific proteins during the parasite life cycle. Okay. And this was published some year ago. But now coming to the other part. So how do tRNA modifications play a role in certain aspects of the parasite biology beyond just regulating the overall translation? So in this case, we identified, and this is a collaboration with Arthur Schur's lab in the Institut Pasteur. We looked at DNMT2, which is an M5C, it's a methylase, and it has been shown to, in other organisms, to be specific for the methylation of C38 in specific tRNAs, and we wanted to know whether this is important in terms of the biology. So we looked at the orthologs in Plasmodium falciparum. There is one gene which has a sort of similarity to DNMT2, but it was actually annotated as a cytosine 5D methyl rather than an RNA transferase. So we looked at this more closely by actually doing a gene knockout. So we just knocked out the gene, and the first thing you see is whether that you knock out the gene or the parasite has a gene. They grow equally under our normal culture conditions. What we also could see is that the knockout has no impact on the DNA methylation. So that's consistent that this gene has been misannotated. It's not a DNA methyltransferase. Instead, what we could show, if you knock out the gene, you impact one specific modification in one tRNA. So it is responsible for the methylation of one site in the aspartic acid tRNA GTC at position 38. So the other methylations that you see is obviously done by different enzymes. So if the question then is, does knockout have an impact on genes which are enriched for the codon that is important for the efficient translation? And so first of all, if you look here in panel B, you would see that the impact is indeed that there is an impact on the protein translation in terms of the knockout. You have some proteins which are upregulated, and then some proteins which are downregulated in the knockout. When you specifically look at the proteins which are enriched for this GAC codon, you do actually see, at a single level, this is, you see downregulations of proteins that are enriched for this GAC codons. At a whole system-wide level, it's confirmed that proteins that are enriched for the GAT, aspartic acid codon, they are not affected at all by this the knockout, while proteins which are enriched for the GAC codon are downregulated. Again, consistent with, yes, the modifications have an impact on translation. Well, I said these parasites grow happily. Does it have an impact on any biology? Well, the proteins that are downregulated are all associated with metabolism. So what this would indicate is that these parasites with the knockout are in some kind of metabolic stress environment. And we know that metabolic stress is a trigger for sexual differentiation in the parasite. So we asked, are these parasites more likely to form the sexual stages, the gametocytes of the parasite, in the knockout than compared to the wild type? And this is actually something you can see even when once in a while you see a frequency of these gametocytes appears to be much higher. When you then trigger gametocytogenesis in these parasites through a stimulation, what you see is that these parasites make much, much more gametocytes than the wild type, which is consistent with the idea they are already sort of prone or ready to just switch. And importantly, the gametocytes that are produced here are, in terms of their ability to transmit, no different from the ones in wild type. So they're not functional defect. They're just, these parasites are just ready to make a gametocyte. And this is also confirmed, by looking at these transcriptional activators of gametocytogenesis, which is a gene called APTG, which is much more activated when you have the knockout parasites. But one other interesting phenomenon is that when you do stress these parasites, you see that the tRNA, this non-modified tRNA, because it is now cleaved. And it leads to these tRNA fragments, which are actually known to be regulators. And this would suggest that the mechanism of this knockout is not only that it leads to decoding of codon or a change in the decoding of codon bias genes, but it also has potentially a secondary role by impacting or generation of these tRNA fragments, which might be the trigger of subsequent developmental processes. So in this part we've shown deletion has an impact on translation, important regulator of different metabolic pathways, and it's linked to gametocyte induction, possibly by the production of these tRNA fragments. Okay, so now, and this is really where we get into the importance for drugs, is what is the role of these modifications in stress response. Again, it's been shown, particularly in model organisms, both in bacteria and yeast, that these tRNA modifications play a role in the translation of specific genes, and particularly in stress. So, for example, these upregulation of this particular modifications in yeast leads to the translation of particular stress response proteins, because first there's an upregulation of this modification, then this leads to the translation of these stress response proteins, and that protects the yeast from this particular stress. This has actually been shown that there are any different stressors lead to different changes in the modification profile of tRNAs, with different, these are 4 different stressors, and you can see each set induces a quite unique pattern of changes in modifications. And what's been shown is if you take a particular stressor and knock out the genes that are associated with the upregulation of these modifications, the yeast grows perfectly happy under normal growth conditions, but when you then stress them with that specific stressor, they die. So it clearly links the modifications to the protective response. Well, is this the case in malaria as well? And I'll just give one example. So we stress the parasites with different stressors, different drugs, from dihydroartemisinin, chloroquine, and other hydrogen peroxide. And again, you see a similar thing, that each of these different stressors induces a different change in the tRNA modifications. And when you look at a particular one, like here, artemisinin, and you look at the modifications, and you then at the same time look at transcription, you see there's a transcriptional change, but there's also a proteomic change. So the drug induces changes in transcription and protein expression, which isn't necessarily surprising. But you can use that data then to ask the question, how are different proteins that are upregulated or transcription upregulated translated? Because you can look at translational efficiency with this data. And what you see is that you have a subset of proteins which have a low translational efficiency and a subset of proteins which have a high translational efficiency. And when you look at this more carefully, you will see that the genes that have a low and high translational efficiency have different codon biases, and specifically codon bias around serine, arginine, and sorry, I can't read this now from here, but at least it particularly affects 4 different amino acids. So how does this associate with the modification? So what we see specifically in the case of artemisinin is not an upregulation of a specific modification, but the downregulation of T6A in response to the modification. So what do we know about T6A? T6A is only found at position 37 of the anticodon loop, and it's specifically been implicated in regulating the specificity of the tRNA. If you in the absence of this modification, it leads to misincorporation of other amino acids. So the question is does the reduction in T6A actually lead or protect against artemisinin? And to address this, we looked at T6A synthesis, which is involves 4 enzymes, knocked out one of the genes effectively. This is just sort of a validation of the knockout, and then ask what happens if you do actually knock down, or it's not a knockout, it's a knockdown. It's an inducible knockdown system. So when you induce a knockdown, what you see as the parasite grows, the level of the enzyme goes down. You can quantify this, and associated is a reduction of T6A levels in the tRNAs. So clearly this enzyme plays a role in the T6A synthesis. What you can also see is when you treat parasites with DHA or expose them, you get also a downregulation of the same enzyme. So that sort of establishes a relationship between the drug and the modification. More importantly, what you also can see is that when you do knock down the gene, the parasite has an increased resistance to the drug. It's not a complete resistance, that's not the case, but it just gives the parasite a bit more opportunity to survive. So, and how would this possibly work? So coming back to this stress, the modification, what does this modification or lack of this modification do? Particularly, it leads, as I said, to the incorporation of the wrong amino acid into a gene, which then triggers would be the protein to be degraded, or this unfolded protein response, which seems to be a key mechanism of how the parasite survives artemisinin stress. It actually degrades defective proteins more effectively, and what this seems to do is, by doing this, the parasite seems to just arrest itself metabolically, and by arresting itself metabolically, it then allows it to survive the effect of the drug. Okay, so just to wrap up here, I think, different stressors induce different changes in the tRNA modification. In the response to artemisinin, it leads to a downregulation of T6A. It affects particularly the decoding of a range of different amino acids, and yes, indeed, it triggers a protective response. And I think this is a mechanism that it most likely has much broader applications for the parasite, as in terms of other stressors, which we haven't yet investigated. And importantly, these are potentially druggable enzymes. They're quite unique. You can think about them if you use this as a combination therapy. If the parasite can't respond to stress, the likelihood of it developing resistance is much, much lower. So just overall, a summary, the tRNA modifications are dynamic throughout the life cycle. They change to the IDC and correlate with codon bias translation. A single tRNA modification can have significant impact on the biology of the parasite. tRNA modifications change in response to stress, and these specific modifications or changes in modifications actually can have a protective response. And now I need to just acknowledge all the people. The project was initiated with at SMART with Professor Dieden's lab, who is a professor at MIT. Ameya and Chiseng were two PhD students who did pretty much all the work that I talked about, but it also involved a collaboration with Arthur Scherf, David Fiddick's group, many other people around the world, and the funding came from the Singapore-MIT Alliance and government funding, as well as funding from the French government. Thank you very much.
Thank you for this beautiful presentation. It was really a very, very excellent beginning of the second day of this conference. And this lecture is now open for discussion. Please ask.
Very nice presentation and great work. So, are these tRNA modifying enzymes, are these enzymes also regulated at the stage-specific regulation, their expression or some kind of, how do they know that at this stage we need to do that and the other stage? So their expression is controlled or some kind of modification?
So the question, because it is how are these enzymes regulated themselves? Clearly, when you think about during the during the sexual stage, during the blood stage, you see a sort of a concurrent upregulation of some of these enzymes as the parasite progresses. So in terms of stress response, we know actually very little how they are regulated. And I mean, in the case of DHA, it would be that the drug itself really triggers a degradation of the enzyme. That leads to a downregulation of the modification. But I think that most likely isn't the case for many other drugs. And we're not yet sure how that sensing mechanism works. Do they get phosphorylated? Are there other activation signals? But the key thing, I think, for what's been shown in others is that these responses happen very quickly. So it really allows the parasite to respond potentially faster than if it's just a transcriptional response mechanism and so on.
It's not a good news that it is very fast modification. Okay.
There's a question here and a question here.
So this is regarding for non-bias.
Yes.
So usually codon bias, it is known that the translation is not, the protein is not changed. But does tRNA modification lead to change in protein sequence, or the protein sequence stays the same?
So in the cases of the classical way, the sequence stays the same, and what happens, you have proteins which have synonymous codons.
Yeah.
And when as a certain modification goes up, the one codon gets preferentially translated. So ultimately, the way I would say is that you have 2 transcripts 1-to-1, or 10 transcripts of each. One that has one codon, the other one that has another codon. In the ones that has the preferred codon, all 10 get transcribed or translated, and the ones with the unpreferred, only 2 get. So you get, it's not a binary, but it's just sort of, these get more effectively translated or faster, because the ribosome can just run through faster than the other. So it has no effect on the amino acid, it just has an effect on how quickly things, or how effectively things get translated.
But usually when tRNA binds, it binds to the coding region.
Yeah.
And regulation is usually done before the gene itself.
Yeah, so this happens at the ribosome level. So what happens is that, for example, a tRNA comes in and it recognizes its codon, and in the case of the wrong codon, it gets kicked out again. So there's a finding the right thing. And when you have a specific modification absent or present, then the tRNA either can only bind to one specific codon, and if the modification is absent, it can recognize all the 4 wobble modifications. So remember, with the tRNA, it's the first 2 of the codon which are really important, and then the wobble position gives more flexibility. And a lot of times tRNA can decode multiple positions, or multiple nucleotides at position 3.
Thank you.
Thanks, and this is very nice and comprehensive talk. Actually, my question is related to the end, which you said, like it induces UPR, unfolded protein response.
Yeah.
The drug, and that's how causing the resistance. So it is known that induction of UPR, many a times it causes induction in the host cell. So, but how basically it is leading to? I mean, because probably the host cell more survival will lead to more production of the parasite, or how it is?
Yeah. So
so
in the context of the malaria parasite, it is in the red cell. So the red cell pretty much is in the context of this inert. There is no so the unfolded protein response actually happens within the parasite. And again, not fully understood, but it's been shown that in thousands of field samples, that it is that upregulation of that unfolded protein response which seems to give the parasite some kind of survival advantage, or and possibly by just shutting pretty much the whole parasite down. Just sort of saying, look, there is a drug, I really don't like it, I'm just degrading everything that's damaged, and I'm just hibernating, or getting into this short-term dormancy. When the drug goes away, I can keep going, and that damage that otherwise would be caused by all these wrong damaged proteins is minimized, I guess. I hope that answers the question.
Yeah.
Other question all the way at the back. And at the front.
Hello. I had the question regarding, can the manipulation of tRNA modification pathways be explored as a therapeutic strategy to enhance drug efficacy or combat drug resistance? Can you tell me about views on this?
Yeah. I think that was a point I was alluding to at the end. I think if you target some of these modifying enzymes and use them in a combination with, so for example, if you think about T6A, because it's downregulated, may not be the right one, but things that are upregulated in response to a drug, and you block that and give that along sort of as a partner drug with something else, you could have potentially a more efficient way of a drug to work. But some of these modification enzymes are absolutely essential. So I didn't talk about those, but there are a whole range of these modifications which are absolutely essential for survival of the parasite. Those you can think of drugging as well, and they themselves then represent a particularly attractive target. There's some of the, in bacteria, some of these TRIM-D, a particular modifying enzyme, is being explored as a new target in sort of for antibiotics, or an alternative for antibiotics. So yes, absolutely.
Thank you very much.
Sir, did this study remove the bottlenecks in the preparation of the vaccine for the
Well, so for the vaccine against, I'm not sure whether they would be themselves a target of vaccine, but when you think about mRNA vaccines that have been explored, the reason why they work is because they actually incorporate quite a number of these modifications that allow the RNA to be avoid degradation and better expression. So you can use it, but they themselves as a vaccine, because they're intracellular, I think they are not necessarily the best target at this stage.
Last question.
Thank you so much, sir, for this wonderful talk. My question is, as you said that DNMT, that is methyltransferase, is inducing the gametocytogenesis, and so can we use combinational therapy so that it will reduce or target the blood stage parasite also and the gametocytes also, one thing. And so there are more than 25 modifications in the tRNA, so which leads to the degradation of the proteins, target proteins, or the instability of these proteins. So should we target these rather than stage-specific protein to the whole parasite cycle protein, like the structural composition or the complex complexes? Please enlighten us.
Well, that's a long question with lots of different sub-questions. So, I mean, the first thing, let's start with the last question. Should you target stage-specific proteins? I think from drugging any treatment for malaria, the first thing you need to treat is the disease. The disease is associated with the asexual replication. Now, ideally, you want to have a drug which also prevents transmission, meaning affects the sexual stages, because then the likelihood of resistance spreading is significantly reduced. Also, as that you prevent the spread of the disease even after treatment. Now, in terms of RNA modifying enzymes, I think, yes, there are enzymes which are important for, maybe more important for the sexual stage, some which are more responsible for the asexual stage. But I think ideally you want to target one that hits both, rather than one for one for the other, because particularly when you think about drugs that target the sexual stage, you have a really tough time thinking about your clinical trial endpoints, so it becomes a nightmare to actually get it approved, because you can't do the proper clinical trial. While if you're targeting something that goes for both, you have a clear clinical endpoint for blood stage activity, and then the secondary endpoint, which is transmission blocking, which you can do in a more phase 3 kind of trial. Very difficult to do in anything beforehand. Thank you very much.
I also have a small question. Are there or can be significant differences in the modification pattern in different strains, different Plasmodium strains?
So that's actually something we don't know. We haven't done yet. These are really difficult experiments to do in terms of the quantification. But you are right. Well, actually, we have. I mean, I haven't talked about it yet. We have a paper which has just been accepted, where we looked at DD2, which is a different strain, and we looked at in the context of the K13, the artemisinin-resistant gene and not. And we do see all the normal modification. Because of the more sophisticated mass spec we were using, we identified, I think, 2 additional modifications which we hadn't seen in our initial. But I think that's more a question of better instrument, higher sensitivity, rather than absence in the upstream.
Thank you very much. This excellent presentation, Peter.
Thank you, Professor Peter. Thank you, Professor Peter, for your informative lecture. Thanks again, sir, for enlightening us with the informative lecture. I hope it will be beneficial for all the participants present here. I request you all delegates and participants to please join us for a short tea break for 5 minutes, and we will again gather here for upcoming session of invited lectures. Thank you.
This is to inform you, all of you, that we are skipping the tea break because of the shortage of time, and we are again running 15 minutes late. So please, all the participants, please move to Hall B and C respectively, and we are starting with the session in Hall A now. Thank you so much.